I'm Jay Kim, coming from Cedars-Sinai Medical Center, and I belong to UCLA as well, and I have a joint appointment at the Harvard Medical School. So today, I will talk a little bit about urinary metabolome project that I am working on. As a translational scientist focused on the urological diseases, so I use a lot of omics approaches to figure out the mechanism of urological diseases, including prostate cancer, bladder cancer, and the interstitial cystitis, which is benign female urological dysfunctions. So today I will focus on the interstitial cystitis as a experimental model to explain about my research approaches using the metabolomics. So there is two grand challenges in omics. The first one is biomarker discovery. Second one is to understand what's the meaning of all the profiling. So to understand that, we need to look for the signaling networks or the pathway mechanisms derived from all the profiles that we got from the transcriptome, genome, or the metabolome proteomics, or different kind of omics. To understand those omics approaches, the omics approaches derive the profile, we also need to apply the bioinformatics tools to extract the central biological meaning from those. So we are focused on combine all of the omics plus computational approaches to understand the biology of the disease, as well as to understand and identify the biomarker. Especially we are looking at the non-invasive bladder, non-invasive urinary biomarkers to apply to the clinical setting more easily in clinic. All right, we focus on urine samples because it's very easy to obtain, and we can get several times a day from the same patient. There is no pain from the patient side of view, and actually there is no very minimum of ethical issues on collection of urine samples. So there is multiple ways to collect urine samples from patient and healthy controls. One is just avoided. We can just collect avoided urine samples naturally. So midstream collection, clean catch method can be applied. The other method is to avoid the possible contamination during the urine collection. We can try to catheterize the sampling as well in the clinical setting. So the urine was considered as a waste of the body, or there is another myth of urine, it's sterile, so we can even eat it or drink it. So, but those two major myths is not correct. It is, we have some evidence for that. Urine can be used as a valuable diagnostic biofluid. And actually, urine has active role by regulating bladder biology. It can affect bladder epithelial cells and result in some dysfunction in the bladder. So why we measure the metabolome to study about the urine? Because as you know, the urine metabolome profile is the last stage of all the signatures. Of course, we will try, in our laboratory, we try from the mutation of genes and messenger RNA expression level profiling and protein expression and the metabolite level and metabolite flux measurement. And we combine all 3 or 4 different layers to extract biology from those profiling. It can be used the phenotyping, classification of patient. Also, it can be used the stratification of the cancer or some disease pathological condition patient from the healthy controls. So there is multi-step to study about the metabolomics as a biomarker. first we need, there is a target analysis is possible. We can target just a few metabolites which we are interested in based on literature search or from other people's product. And second, we can try the unbiased metabolite profiling. And so it can be used to classify the sample. This diagram shows the ideal workflow that we are trying to. The first, we do the sampling from the patient, and it can be from cultures, from the different stages of disease sample, and we use the high throughput extraction and analysis, and we do the data processing using the bioinformatics as well as more comprehensive integration, data integration approaches. And we know we will get the output correlated to the disease state and profiling. And we can use this information to treat patient. And we also use this information to monitor patient response to the drug treatment or therapy treatment in clinic. So there is a couple of technical issues in using the metabolomics analysis. The first one is we need to choose analytical techniques, NMR or mass spectrometry-based metabolites, which is better. As you know, the both has advantages and disadvantages. For example, NMR has limitation, but it has also advantage, a lot of strong advantages. Mass spectrometry-based metabolomics is sensitive than NMR in general, and there is more database available to extract biology from the profiling. And we also need to consider we should try the targeted metabolomics analysis or non-targeted metabolomics profiling. So it's good to do the unbiased method, but sometimes we are in the middle of a trap, too many data that we can digest at all. So we need to consider all of those. And we also need to consider the comprehensive data processing and the metabolite identification. Sometimes, after we got a list of NMR spectrum, for example, we ended up where we cannot annotate all of the interesting spectrum. So we don't know what those are due to the limitation of database that we have now. So there is a lot of efforts, international effort, is going on to enrich the metabolomics profiling. So we are working on that. So to simplify all the procedure, for example, in my case, I'm interested to get the signature to stratifying interstitial cystitis patient from the healthy cultures. So we got the urine samples from two groups, IC patient group and the cultures group, and we do the sample preparation. Most of IC patients, over 80% of IC patients are women. So we don't need to worry about prostate massage kind of procedure before we collect urine samples. But in case of men population, to study the IC profiling, maybe we need to think about the different procedure to collect urine samples. And we do the NMR, and we can do the CC or LC based mass spectrometry and pre-processing, and we identify some possible profile that is different, that are completely different from the culture. And then we do the statistical analysis, and we annotate, and we figure out possible potential IC biomarkers. And we validate using different platforms. For example, if we got some idea used based on the NMR metabolomics, maybe we can try to validate those signatures can be found using mass-based metabolomics analysis. We can try that in different cohort, international cohort as well. But there is analytical challenges that we really need to consider. because as you know, there is a wide variation in the ionic strength, pH, osmolality, particularly under the condition physiological stress, and medications affect a lot on the metabolic profile. This is one of the examples from a review article from my laboratory, the Metabolomics Insight into Pathophysiological Mechanism of Interstitial Cystitis. In this paper, we reviewed the literature's available so far, and we suggested some the future roadmap. So recently we got interesting data from the urine profiling from the IC patient and the control, age and gender matched control groups. So I want to talk a little bit about that. So this is a major question that we had, as I mentioned before. The first one is, what's the differences of the patient sample from controls? And the other one is why it is different. The first one can give us the diagnostic tool variable, the biomarker. And second question can give us the biology and the mechanism and give us the possible strategy to target disease. So interstitial cystitis is very interesting and not appreciated so much so far. So this is chronic syndrome of unknown etiology. So there is no mechanism understood so far, and very common bladder disease among older generation, more than 1 woman out of 77 people in USA. So it affects quality of, the symptom is like that. So there is always frequency, urgency, and nocturia, and the public pain during the urination. So it's because of this uncomfortable and pain, patient cannot have the qualified life. They always think about where is restroom. So it really limits their social activity. So it's a really big problem for the public health, affect the quality of life and productivity, work performance, so public health burden. And there is only one FDA-approved oral drugs available for patient. So it's the problem. So, but it doesn't affect most of the patient, and about 20 to 30% of patient has benefit, but most of those has the recurrence pretty quickly, like 2 months later, they can come back to the clinic to get another treatment. So we need new medications for ICE patient, and we also need objective and clinically relevant indicator for ICE patient. We had another, maybe 10 years ago, we discovered urinary biomarkers for the ICE patient. 95% of patient has this bioactivity of this small peptide. It is called anti-proliferative peptide, APF, and it was abundant in the patient compared to control groups. So we follow up the bioactivity of this small peptide. It's very mysterious peptide. It's 8 amino acid peptide, small fragmentation of frizzled 8, which is the receptor for the Wnt signaling. But it's not really related to the Wnt signaling pathways. So using this APF small peptide in vitro system, we wanted to look for what's the mechanisms. So we found, based on various different approaches, we found APF, this small peptide found in urine samples of specifically in IC patient, can suppress P53, which is important transcription factor, and decrease the cell proliferation and cell cycle, and related to the interstitial cystitis. And we also tried the proteomic approaches in vitro system. We treated the cells and compared to non-treated cell bladder cells. We found some interesting pathway was enriched. It means downregulated or upregulated in the APF by APF treatment. Those was mainly beta-catenin network and P53 and MAP kinase pathways. So we confirmed those informations in the separate validation experiment using the Western blot analysis. It was published in Molecular Cell and Proteomics a few years ago. And then we wanted to go to a more in vivo system human setting. So we got this in vitro cell line system proteomics analysis, and we tried to combine those information with in vivo human setting transcriptome data, which is publicly available. We integrated those two data completely different, separated, independent. One is in vitro system, the other one is in human system. And we found inflammation or immune response-related pathway was very much enriched in the IC conditions, which was pretty consistent in the pathological observation in patient actually. So it was interesting. We could have published several papers based on this data. And here, I need to mention about that. So when we look for the possibly available database from the literature or the gene bank or whatever, or possible research, we realized for this disease, there is very limited omics data available, actually. Not even high quality, because when we look at the number of the patient samples, like N5 is the maximum, something like that. So we wanted to expand our observation into larger cohort to validate our the observation.
So you have 2 minutes left.
Yeah, sure. So we got samples from patient, and we did animal-based analysis, and PCA and the OPLSDA, the statistical analysis showed a beautiful stratification of the patient samples from cultures, which was great. And this is heat map also showed the candidate 140 NMR spectrums, which is spatially associated to the IC patient compared to culture. So we wanted to identify those spectrum, and we realized the tyramine 2-oxoglutarate can be the target or the candidate IC-associated candidate biomarker from urine samples. This is summary. So we know a little bit about the mechanism of this disease based on in vitro setting experiment, lab-based experiment. Also, in vivo setting, in human setting, we also identified some possible biomarkers derived from urine samples using the metabolomics profile. So using the another layer of omics approaches and computational analysis, we will get the mechanism and the possible targets for the therapeutic treatment. I am in the MAP network, which is NIH, NIDDK sponsored. It's a UO1 consortium. It's first cycle is almost ended last year, and the second cycle is just started. The UCLA and the Cedars-Sinai here involved as a discovery site. This is my acknowledgement. NIDDK and DOD, also the Interstitial Cystitis Association pilot grant helped our research very much. Thank you.